for someone who is selling or buying a dog. But I know in the show ring you won't accept long hair. German Shepherd, we don't accept them either. In German Shepherd, we don't accept these dogs, also because they have more problems with the skin. They have more dermatitis and that's why in German Shepherd we don't like these dogs to have too much hair. Interesting book to read is Genetics of the Dog. It's really difficult. But if you are interested or you really want to know a lot about genetics, then this is a nice book to read.
I've only got to page 1. This is I'll go to page 1 in 2 years.
Okay, something very important that you should understand is the biochemistry of heritability. I won't go too deep in details. But it's very important that you understand this, to understand what we are doing when we do a genetic test on a dog. You see here the DNA, all kinds of images of DNA. I think every one of you, if you are a breeder, that you know about the structure of DNA. Here you have the genetic code, and it's in all the dogs. It's about the same, but there is also some variation, which makes the difference between one individual and another individual. That's what Crick and Watson have found. And what's the most important thing you have to understand is that DNA is the genetic code of proteins that are doing the job in your body. They're doing everything. And you can compare it the best with, you have a mole, so this is the DNA, and you make an apple out of this mole. This mole will remain the same, and you can, once this apple is finished, you can make a new one. That's what happens in the body. There is the DNA encodes for the production of protein every second, every millisecond. All these apples or these proteins are used in the body to have a good functional body. It's the same with this here for little soldiers. You have also a mole, and you can make 1,000, 10,000, 1 million little soldiers just using one mole. Yeah? If the mole is not good made, same comparison, if DNA has a mistake in it, if the code is not correct, then you may have bad proteins that do not function properly, and then you can have an animal that's diseased. You can see that it's diseased, or it's not diseased, or it has, for instance, a bit worse character. It has some higher laxity. So these are the proteins that are the result of DNA. If you have, for instance, a mutation, then sometimes you have a protein that's much bigger than what it should have been. So if it's bigger, it will not function that good. If you have a small entrance, then you can go in with this kind of proteins or amino acids. If you have small entrance and you have a big protein or amino acid, then this amino acid or protein cannot enter the room anymore. So it's blocked and cannot do its function. All nice images just to show you how complex it can be to construct out of DNA proteins. These are all stuff you need to do this kind of protein production. But it's very crucial for a good functioning body that the genetic code, the DNA, is translated properly in the good protein. What can happen? DNA is replicating all the time in your body. Also when there is, if you have eggs and sperm formed in your body, or in the body of your dog, then DNA will be replicated every second. And sometimes it can happen just like monks that copy books after books. Sometimes they make a little mistake. They write a little mistake in the book. That's what can happen in DNA as well. While the replication is going on, there are some mistakes in the new copy of DNA. And that's what we call the mutation, DNA mutation. This, for instance, here, there should be always an A at that place in the genome, but by mistakes it's replaced by a C. So you have another word or you have another sentence. And if you change a letter in some words, then you can have that it's a word you cannot understand anymore. So it's the same for DNA. In some cases you have faults written down in DNA that cannot be used anymore for the construction of the protein you need. And it's these places that will be looked up when you do a genetic test. So if you send in a swab and you ask, can you test the dog for this disorder or this trait, color, or long hair, then the laboratory will look if on this place you have an A, and if this is the normal, and you have 2 times an A, then you will get a result that the dog is free because he has no mistakes on the DNA. They can find that in one of the words you find an A, but in the other word you find a C, then they will say that your dog is a carrier of this mutation. He will not have a problem in cases it's a recessive disorder, but he can transmit this C to half of his puppies. If you combine this with a female that has also one mistake in her DNA, then she will transmit it also to 50% of her puppies. And in 50% or 50%, you can have the opportunity that C meets a C. And then you have 25% opportunity of chance that your puppies will have 2 times a C and that they will be affected.
So, just something very interesting here for breeders. Mutations are possible, but mutations are not necessarily acceptable. Mutations, that's why there is a specific breed standard that often just describes what you see on the outside. But we've always, in cattle with horn shape or something like that, there is a standard. The mutation will come up, it will fall outside, and it's on the stamp and the standard that you set for the breed initially to decide, is a possibility acceptable or is a possibility not acceptable.
Yeah, but it's also important to know these genotypes to have the right combinations again.
Yes.
Yeah, but also in such a way that if you allow certain combinations, even allow in some cases you even have to allow that affected puppies are born.
Yes.
If you have a big prevalence, but always keep in mind that with every generation the prevalence of the incidence of the mistake in the DNA in the population will lower as well.
Yes.
Yeah, yeah. So that's the general idea. Yeah.
Except if you have a dominant genetic.
If it's a dominant, yeah. But then you will see it immediately, the dog will be affected. And then even you have to think, what's the consequence of this dominant mutation? Because sometimes it can only be that it's a bit of a floppy ear,
Yes.
and that the dog will not suffer from it.
Yes.
But if it's a dominant mutation and the dog will die within 6 months, or at the age of 2 years, maybe you should think of not using it.
Yes.
Or maybe you don't find a good combination. Maybe it's better to say you will not find a good combination with a dog. But anyway, it's important to know for 2 dogs that look very similar, that one has 2 A's and is therefore completely free. The other, he looks the same, but he's a carrier. You should know it, and that's only thing you can do is using genetic testing. So these are all different types of mutations that can happen. One is having a huge impact, others have less impact. But that's not very important. What I want to say to you, and it's the following of my explanation, is that you can have simple inheritance and you can also have complex inheritance. For the simple inheritance, so this monogenetic, recessive, dominant, intermediate, the methodology to deal with these disorders or traits is through genetic testing. And for the complex diseases, that's what I will tell after Rijn has his little speech. It's complex means estimated breeding values. So I will make already very clear. Monogenetic, simple inheritance is identifying using genetic test. Complex will we do need the breeding values to find the animals that have more mutations than others that are not liked by us. So simple inheritance, just a small example, this is in calves, it's just a pathway that helps animals to clear off ammonia that's produced in everyone's body. And there is one protein you need to make ammonia to become urium. So if you haven't been drinking a lot, you go to the toilet, your urine will be yellow. The yellow color is from urium. So you see here a protein which the DNA code looks like this. You have a long word, but in the middle of the word you see each A. Yes, this is the wild allele, this is the normal allele, and there's just one little mistake. The C has been written as a T. So the monk has been writing, someone said, and instead of a C he wrote a T. The result of this mistake is that this protein is no longer made in the body of this animal. This means that your ammonia cannot be translated or transformed into urium. So this means that ammonia will increase in your body. You know, if you use ammonia in your kitchen or your floor, you will have burning flags. Yeah. If you drink it, you're dead within 10 seconds. So these animals will be intoxicated after a while. Within 3 to 5 days they will die, because they are just intoxicated by ammonia. It's just a genetic cause of intoxication by ammonia. So you can use a genetic test to see whether this animal itself, when it is born, is a sufferer. Then you immediately will know this animal will not last for 3 to 5 days. But you can also check, for instance, the brothers or the sisters of these animals, if they are carriers or if they are normal. Yeah? So that is by a genetic test. So these are the monogenetic diseases. This is an example of colors, brown colors in the eye, blue, how it's inherited. Brown is dominant. So you see that if you have blue eyes like I have, my parents have both brown eyes, this means it is immediately a proof that both my parents are carrier for blue, even though they have no brown eyes. Or it should be that my father is not my father. This is something as a breeder you can do yourself, is make some pedigree. If you have a male you use a lot, you have some females, you can put in some traits, and then if you have a certain trait that you want to follow, you can yourself estimate whether it's a recessive, dominant, or if you cannot find any clue on the inheritance, then you probably have to deal with a complex inheritance. And then you need some other people to help you. Okay? Just some examples of pedigrees, not going into detail. So you have here an affected animal or an animal with a certain trait. For instance, you have a white Boerboel, suddenly born in your puppy, with your puppies, and then you can try to figure out where this white comes from. You have to make such a kind of pedigree. Here, 4 calves that were affected in the Netherlands, in Denmark, 3 calves, and then they looked for the parents, and then they found that at a certain moment they found a common ancestor which was believed to have been the source of the genetic mutation that's popping up here. It is a monogenetic disease that's popping up here because these animals are inbred on A and on B. That's how autosomal dominant looks like. Every generation you have some affected animals. Recessive means that you have out of 2 carriers, 1 free, 2 carriers and 1 that's affected. So it's 25, 50 and 25%. That's stuff you know. You can also have genes that are on the X chromosome. They are dominant or they are recessive. The best known example is hemophilia, so bleeding disorder. It happens in humans, and I know it's also in German Shepherds. I don't know if it's in Boerboel.
Not the ones we tested so far.
But we will know within some months, if we have done a test on a lot of animals, then we will be able to tell whether it's in the Boerboel as well or not. Queen Victoria was a carrier. She has a lot of sons that were affected. You know Queen Victoria?
No, never met her.
She died in 1900.
But they're not Europeans. I think she's more.
No, it's South Africa. Are you not the British Commonwealth? Or no? So, in cases of simple inheritance, it's possible to screen the genetic mutation with the help of a simple genetic test. So, I told you all these stuff just to show you what effect it can have. It's just one simple mutation, difference between big and small, hypermusculated. Even here the colors, yellow, black, just because of one simple mistake in writing. This is more an example of an
Nice.
we aimed at producing a child looking as good as Marilyn and being as bright as Einstein, but it was the opposite. We had bad luck. So this is what we have seen. Yes, this is very important because we have a nice example for the Boerboel. That's why I was explaining to you. You can have a genetic test that looks whether the C has changed in A. Then you get a result. Okay, your dog is a carrier because we found once a C on one of the alleles, and in the other we found an A. And yet this dog has long hair. Yeah? So you think he's a carrier. He cannot have long hair, which is a recessive trait, but we should look at other places in the same gene as well, because you cannot only have a mutation at this place, but also at this place and also at this place. So what can happen is that the dog who had long hair had the mutation at this place in his one allele, and in the other he had a mutation at this level. So in fact, he's not a carrier. He's a carrier of A, he is a carrier of G, but being carrier of A and carrier of G makes that you produce bad proteins in both cases, and then you have also the dog who has long hair. So that's the tricky of genetic testing. If you get the result he's free, and you only test at this level, then he is free for the mutation from C to A, but it's not telling you anything whether he's free of the G or the T mutation. So the conclusion of this is that you have to test not only for the A, but also for the G and also for the T. And if they are normal in all cases, then you can say he's free and he can be carrier of this, or carrier of this, or carrier of this. But in all cases where all these mutations come together, you have in fact a sufferer. Okay, that's a little bit more difficult. In Alzheimer's disease, they have looked some genes, and they found that one gene that's very important, they found 165 little mistakes that all help you to become Alzheimer's disease, dement at an age of 70 to 80 years. So then you have to look for all these mutations to be sure that you will not become mentally ill at old age. Okay? That's what I have found, that there are up to 100 genetic tests for about 140 diseased or mutated genes, which means that indeed there are mutations or tests that look for different mutations in the same genes. One background is that we are not always sure if they are correct, if they are described in scientific articles, and you cannot always be sure that they can be tested in the laboratory, especially not in a routine laboratory. So that's what is done now in the Van Haringen laboratory, is all the tests that have been described, we will try to do them in the laboratory. Sometimes the conclusion is it's not described properly by the scientist. Was it a mistake or was it on purpose? We don't know. Or it's such a huge mutation that it cannot be done in the laboratory. And sometimes we find also that, okay, the mutation was described well by the scientist, but he's described this mutation in a certain population in his laboratory, and probably this mutation is only segregating in that population, and in the outside world this mutation does not exist. Okay, this was a bit about genetic testing. Rein will now show how you can use results of genetic testing as a help to do some proper breeding combinations. Okay.